Hey guys, so this is going to be chapter 3, and we are going to do the first two sections of chapter 3 for Bio 1010. Let's get started. We talked about in the previous chapters, we talked about just biology in general and the different starting points of learning about biology in the first chapter. And then we talked about the levels, and we talked about chemical level already, and we talked about chemical bonds and molecules and macromolecules. And now we're moving on to cells. And if we remember from the very first chapter, chapter 1, that cells were the smallest living unit that can do all those properties of life that you had to memorize before, like growing and reacting to its environment and all of those things that are properties of life. Cells do those. So we are going to talk about that in this chapter with cells. If you are studying cells, it is cytology. Study of cells is cytology. If we were looking at lots of different cell types in a tissue, that'd be histology. So how do we study the different cell types? First, we have this picture here of the different cell types. So the cell is going to be constructed based on its genome, is how the different cells will become what they are when they are growing. They're growing and developing based on the genes inside of them. So we are coming from our DNA, and that DNA inside of the one zygote cell, when the egg and the sperm came together, created all of the different types of cells in our multicellular body, which is just mind blowing in its fascination on how that one cell can become all of these different cells that can make, eyes and hair and all of that stuff. It's super cool. So cells can differentiate. So we can see cells here. These are sinus cells. Uh-oh, my Apple Pencil's dead. These are sinus cells in pink here. So if you're looking at your sinuses, if you took a slice of that and put it under a microscope, that is what you would see are these pink cells. My mom actually had sinus surgery just this week, and so I got to see way too much of the inside of the sinuses. So these are sinus cells in pink on the far left. If we were checking out plants, plant cells have a cell wall, which makes them pretty distinctive when you look at them under the microscope, versus an animal cell. Animal cells have a cell membrane, but no cell wall. Plant cells have both. They have a cell membrane and a cell wall. So they look rectangular shaped here in these blue looking, still not going to work, in these, there it goes, in these blue looking ones right here are onion cells. So if you grabbed an onion, sliced it open, and put it under a microscope, you'd be able to see something similar to this. It'd look even better if you added some stain to contrast dye on it, and it'd look really cool. If we were in a face-to-face class, you'd be able to do a cheek swab and be able to see your animal cells, kind of similar to these here. And then our very last picture here is C, and this is a bacteria. This is Vibrio tasmaniensis, and it is, there's lots of different Vibrios out there. Vibrio is the genus. I don't know exactly what this one is, but there's lots of different Vibrios out there, like cholera is a Vibrio. And so they are bacteria, and bacteria are in general, we're going to talk about size later on in this video. Bacteria are in general smaller than animal cells, smaller than eukaryotic cells like plant and animal cells. So here's our little bacteria. Bacteria up here. This guy is the plant, onion cells, and this is sinus animal cells up here. How do we study them? We got to look at them under a microscope. So we didn't have microscopes until the last couple hundred years when they were finally invented. It kind of started out as like Galileo and his scope, but there are some other, if we went into med micro, we would talk about all the different history of microscopes. In this class, we're just kind of skimming over. So we're going to talk about the different types of microscopes, and there are three basic kinds that I want y'all to be able to kind of recognize. And we're going to start from the lowest magnification to the highest kind of magnification. So low magnification is going to be a dissecting microscope. And so that's like these guys right here. That's a dissecting scope. If we were having this 1010 class in a face-to-face lab format, you would be able to use a dissecting scope. When I tell you about slicing the onions and putting them under the scope, you could see that. If we used Alodia algae and we picked a leaf off of it and put it under the dissecting scope, it would look like these guys, except if it was Alodia, they have pigments and you'd be able to see the green in the chloroplast, but it would look just like that under this dissecting scope. So dissecting scopes are pretty cool. They are going to have this just flat stage. And they're going to have a light source, usually from two different places. There's usually a bottom one underneath a little light bulb under here. And then there's usually a top source over here that shines down, and you can usually go between the two different light sources, bottom only, shining from above, or both at the same time. How you focus it, you're going to look through the eyepieces, and then you are going to turn the focus knob up and down, and that is basically your sole focus. So just moving it up and down to that object getting in focus. For our dissecting microscopes, they don't get too great as far as magnification, but they're still much better than the naked eye. Magnification is a term I'm going to want you to know, and that is just basically how um, large we can make this look, how the image is enlarged. Um, Basically zooming in, zooming in on your photographs. That's magnification. When we enlarge it and see it as, X number of times the naked eye. So if we looked through a light microscope, that's this guy. This is a light microscope. This one is 2, so it's a compound binocular because there's 2 eye sources here. To put both eyes there, so this is a compound binocular light microscope. If you were in a face-to-face lab, you would need to know that for the lab practical. And with that one, there is going to be 10 times magnification. Just looking through those oculars, the piece that you look through with your eyes, is 10 times. And then depending on what lens, and there's different objective lenses, there's one here, there's one here, and there's one here. There's another one usually hiding in the back. There's usually 4. But some microscopes only have 3, and those objectives change, and they range in magnification and go from low at scanning all the way from scanning, low power, high power, and then oil immersion if you're lucky enough that your microscope has that, and that will get you up to 1,000 times. 1,000 times what you would see with your naked eye. That's as high as a light microscope can go, is 1,000 times magnification. There's another term that you're going to need to know, and that's resolution. And with microscopes, resolution is how well can you see the image? Can you distinguish between 2 details that may blur together if it was poor resolution, but if it was better resolution, you'd be able to see that those are in fact 2 distinct different spots. If we were talking about resolution as far as televisions, you want to get a new television or a new uh, computer, and you're looking at the resolution on that screen, right? What kind of resolution does it have? Does it have the retina display with X number of pixels? Um, that's similar to resolution, being able to see the fine details and distinguish it between two points that are next to each other. That's resolution. Basically, kind of your focus is resolution. So you need to know those 2 terms. Magnification is zooming in, blowing things up, X number of times what you see with the naked eye. And light microscopes go up to 1,000 times magnification of your naked eye, what you're looking at. And resolution is the ability to focus, the ability to separate out 2 structures that may be next to each other and distinguish those. That is resolution. That's seeing the details is resolution. So here is our dissecting scope. This is our compound binocular light microscope, and we're getting higher and higher in our level of magnification. Our last stop, this is an electron microscope. These are they're not the highest, but out of this PowerPoint and out of this description and in this textbook, this is as high as I'm going to ask y'all to go. Electron microscopes are going to beam these little guys against or through a structure, a cell, And so if we were talking about beaming the electrons and it was going to scan the surface of a cell, if you saw this guy, for example, we can see the surface all along the edges of this little Vibrio. Here he is. Here this one is. We can absolutely see the surface features. That is a scanning. Here it is, scanning electron microscope. Scanning the surface, and it may be, you may see them abbreviated as SEM, scanning S, an electron microscope, SEM. So they can come in scanning electron microscopes where the electrons are bouncing off the outside of that object, of that bacteria. Or we may see them as transmission, TEM, and those guys are going through. So we will see pictures of things going through. All of these electron microscopes, these pictures send back. They're computerized, they're black and white, and we, Photoshop in the colors on them. So if you see teeny tiny pictures, Vibrio is not naturally, the brown color is added in there to show those features. And same with transmission microscopes. Those colors are added in to show the different structures. So if you were looking at, the mitochondria on the inside of a bacteria, not on the inside of bacteria, on the inside of a eukaryotic cell, and you're looking at the mitochondria, that's going to be on the inside of the cell. So it's on the inside of the cell. It has to be a transmission because transmission is going through to look at the internal structures while scanning stays on the outside. So these are our three different types of scopes that we need to talk about. Again, dissecting, light microscopes, and electron microscopes are our three different ones that we talk about in this textbook to study things. If you decide to go on to MedMicro, you will learn about a whole lot of more very complex microscopes. But this is what we're covering in Bio 1010. Parts of the microscopes. If you are in a face-to-face lab, and you have the upcoming lab practical, you're going to need to know every single part on this scope. Fully online Bio 1010 is going to be totally different. It will be, the software that you're using. But face-to-face ones, you need to know these. So I kept this in here just so y'all know the structures too. Just one more source of information on the different structures and parts of a microscope. When I'm talking to y'all about oculars, that's these guys. When I'm talking to you about the objectives, that's going to be all of these that are here. And there's 4 in this picture of this microscope that I took right here. Some microscopes, depending on the class, may not have that oil immersion, may not go all the way up to 1,000 times. But these are all the parts, just for reference, of a compound binocular light microscope. Talking about those electron microscopes versus light microscopes, here is a picture of bacteria, and they are so small. They're so small. Look at all these little guys. Nice little guys. This is salmonella. And it's very, very small on this light microscope. We said, remember, light microscopes can only go up to 1,000 times what you see with the naked eye. While electron microscopes, they can go much, much higher on this view. So here is our bacteria, and this is at 25 micrometers. And here, when we look at salmonella again, here is the actual individual salmonella, and we can see it trying to get into, salmonella are bacteria. These guys are trying to get into a eukaryotic cell. And remember, you and I, when we learned about this in the previous chapter, the 3 domains, we are eukarya, so we're in a different domain. So here's a eukaryotic cell. Here's this human cell, and the salmonella are trying to get in. So these guys are trying to sneak into, your gut biome. Maybe you ingested a bad strain of salmonella, and it's trying to get into your intestines and make you sick. This is a picture of it, and we can see this in action, trying to see these tiny bacteria cells invading in the larger eukaryotic cells, and we can see this using an electron microscope. So here is our scanning electron microscope looking at the external surfaces of these 2 cells. Well, this is one cell, This is eukaryotic cell. And then here are, 1, 2, 3. It looks like there's maybe 4 or 5 of them trying to sneak in there. So lots of different salmonella that we can see under an electron microscope. So electron microscopes are super cool. We do not have them at Georgia Highlands, but at some of the other maybe bigger schools, maybe they have them, but we don't have those here. We have light microscopes, and this is about what you would see in our face-to-face labs. We said study of cells, cytology, and females in the class, once you become a certain age, you go and make sure that your cells are not cancerous. And so you'll go in, it used to be every year, and now it's every other year, I want to say, to go in and make sure that your cervical cells are doing okay, and that they will check them with a pap smear. So we can see here are uterine cells, and these are, normal cells. We've got these regular nucleus right here, regular old cell membranes, normal size cells, everything's fine. And then here, we have these enlarged nucleus. These cells are not normal. These are abnormal cells. This is an abnormal pap smear. And so we would need to detect if these are cells are normal or not. And if they're abnormal, what's going on with these cells? Are these cancerous cells? Is this potentially uterine cancer? So this is a great way to be able to use cytology to determine what's going on. And is somebody healthy, or does somebody need to get checked out? And if we are talking about uterine cancer, if we're talking about cervical cancer, that is associated with HPV, with human papillomavirus. And now there is the vaccination out there that you can get. And at least 2 of my kids, I think, have had it. My youngest, I don't think, is quite old enough to get it yet. But my 2 boys have gotten the HPV vaccine. And granted, they don't need to worry about cervical cancer because they're boys, but they may pass it on to somebody else. They may pass on HPV to somebody else. So vaccinations are your friend. And so my boys are vaccinated, even though they don't need to worry about that. They're vaccinated against those certain strains of HPV that are known to potentially cause this. So this is a great way to be able to look and study cells and see, is something healthy or is something abnormal. Cell theory. We definitely need to know cell theory. It's going to be a test question, at least one test question. And there are 3 tenets to cell theory. Know them, love them. Cells are the building blocks of all plants and animals. Absolutely. We said that cells are the smallest form of life that can perform all the properties of living organisms. They are the building blocks. So you can't have all the properties of life in a one unicellular thing like that Vibrio, like that bacteria. It does all the properties of life. Multicellular organisms are still built off of cells. They are still the building blocks. Plants, animals still need cells. All new cells are going to come from the division of pre-existing cells. You can't just magic a cell out of thin air. It has to come from somewhere. So it's got to come from mitosis, from meiosis. We have to be able to get a cell from a pre-existing cell. And then the last one, those cells are the smallest units that perform all the vital functions, all the properties of life that we talked about. So know these 3 tenets, please. They will be on your test. If you have questions about cell theory, let me know. That's going to finish up our first section of chapter 3. Lecture quiz. Okay, next section is going to compare prokaryotes and eukaryotes. So prokaryotes, remember, in our phylogenetic tree of our 3 domains, there were archaea and bacteria, and archaea was a domain, bacteria was a domain, and eukaryotes, is the 3rd domain. So we're going to talk about prokaryotes first, and then in the next section, 3.3, we bust out into eukaryotes. So prokaryotes, what do they look like? Here is our kind of generic prokaryote. They are going to be, in general, these guys are single cell. They are unicellular little ones. So we're not going to see a whole bunch of multicellular guys. These are, in general, unicellular, single celled bacteria, archaea. They are not going to have organelles that have a membrane bound thing, like nucleus has a membrane around it. We're going to see it in the next video. These guys don't. There's no membrane around here. What they have is their DNA, just like us, but their DNA is housed in just this area, this designated area, and it's called the nucleoid. Nucleoid. So don't get it confused. The nucleoid is going to be found in prokaryotes. Eukaryotes have a straight up nucleus, and it's membrane bound and it holds all the chromosomes inside. Prokaryotes don't have that. They don't have a membrane that holds all the genome inside. It's just this kind of area where the genome hangs out. So all of their DNA is in the nucleoid region inside of a cell. Their DNA is going to be a giant circle, by the way. They're going to have circles. So they're going to have giant circle of DNA where prokaryotes do not. Prokaryotes is a circle. This is prokaryote. You can't see the R. Prokaryote, circular DNA. Eukaryotes. When we get to mitosis, and hopefully you've talked about mitosis in high school biology before you got here. But just in case, hopefully you've seen a picture of chromosomes, and they are typically linear, and oftentimes you see them in mitosis, so they've got their sister chromatid attached on. They kind of look like little bow ties, but we have linear chromosomes. So our DNA is linear, while prokaryotes is circular. And later on, you're going to make one of these, which is going to be cool. It's called the Plasmid Project. You are going to make your own little circular DNA plasmid out of paper strips. And there's a whole video. It's a whole project. It's going to be at the end of the semester. But you're going to make your own circular DNA strand called a plasmid that is just like a prokaryote has here in this picture. So let's get back to the picture. We have the DNA. It is circular. It is housed inside an area called the nucleoid. It sits inside of cytoplasm, cytosol, the liquidy stuff, in the cell. So it's floating around in that area. Other things floating around in the cytosol are going to be ribosomes, because we said with central dogma, you are going to go from DNA, and we're going to go through this later on in another chapter, and you're going to make RNA, you're going to make messenger RNA, and it's going to meet up with ribosomes, because you can't have amino acids, which are your building blocks for proteins that we learned about previously. You can't build proteins without ribosomes. So eukaryotes have ribosomes, prokaryotes have ribosomes. Everybody has to have ribosomes. So there are ribosomes floating around all in this little prokaryotic cell. So we have our DNA. We've got our ribosomes floating around in there. They are going to be encased in a cell membrane. Every cell has a cell membrane. Every cell. Every cell has to be able to protect itself and make sure that what's outside can't come inside without some sort of barrier, and what's inside can't just fly around outside. You got to be selectively permeable, deciding what comes in and what goes out. So every cell has a cell membrane, every cell. Not every cell has a cell wall, but we've got this cell wall here. This is our typical generic prokaryote. It's going to have a cell wall. And then there's going to be some optional features on this prokaryote. Not every prokaryote has a capsule. Not every single one of them does, but oftentimes are pathogenic, and pathogenic bacteria are the bad guys, the ones that make you sick. Pathogens typically do have a capsule, and that capsule helps them stick onto surfaces like your intestines, where we saw that salmonella attaching onto the eukaryotic cell and trying to sneak its way in. Capsule helps it do that. Capsules help them be sticky and stick to surfaces like the plaque you might have on your teeth. Hopefully you brushed your teeth today. But in case you didn't, or it's been a couple hours, you've got plaque growing up, and that's different bacteria. And those things may have capsules that help them stick to the surface of your teeth and attach on. Capsules also help them be really big. So when your immune system, your white blood cells come along and try and phagocytize them, try and eat them, that capsule makes them so big that the white blood cells are like, oh man, I can't even touch that. That's way too big. And they'll pass on it and they can't eat it because of the capsule. So they may have the capsule, they may not. It's optional. Other optional things that prokaryotes may have. They may have a flagella. Not every bacteria has a flagella, but they may. And that flagella can help them get around. They may have one, they may have multiple. It may be on one end, it may be on both ends, it may be all over. They may look like a hairy beast. but they may have flagella, they may not, optional. Pili. Pili are optional. Sometimes you'll see them have reproductive pili. And that is basically like a really long lasso that they will go and lasso another bacteria and they will swap genetic information back and forth. And we're going to talk about that in another video too. But that is pili, another may have it, may not optional kind of thing. What they always have, they're always going to have DNA. They are always going to have a nucleoid region in here. They are always going to have ribosomes, got to have them. And they are always going to have a cell membrane. Cell walls vary. They may have a thick one, they may have a thin one. Some of them don't have them. They may have a capsule or not, they may have pili or not, they may have flagella or not. But everything with a star are structures that prokaryotes are going to have. They all have these structures. I stole this from my Med Micro textbook PowerPoints. So it's a whole lot of detail in here. But if you were trying to compare eukaryotes, the third domain that we're in, versus prokaryotes with our bacteria and archaea, it breaks it down, talking about how cell size is in comparison. In general, eukaryotes, plant cells, animal cells, fungal cells, those guys are bigger than bacteria and archaea. It says that, we have a nucleus where prokaryotes do not, no nucleus. How do they divide? They're going to divide by a process called binary fission. We'll talk about that later on. We talk about cellular reproduction, while eukaryotes are going to use mitosis and meiosis. We have eukaryotes. We are called eukaryotes because we have membrane organelles, while prokaryotes do not. So there are some basic and more detailed information in this table, but I wanted you to have it as a comparison, prokaryotes versus eukaryotes. Cell size. Here's that cell size slide I was telling you about that compares the two. So here's our person. Here's an ostrich egg, chicken egg. If you had eggs this morning, I did for breakfast. If you did, there's your chicken egg. Frog eggs. If anybody has seen tadpoles and frog eggs out there, again, I think I told y'all in my last video that I have a backyard and it has a pond in it. And every year we have frogs. I went to clean it out just last week to get some of the leaves out of the bottom of our little pond, and there was tadpoles in there now. So we have tadpoles in there year round, but frog eggs that you can see with the naked eye are small, and so you can see, relatively speaking, they're about 1 millimeter. So if you busted out, your childhood ruler that has metric and imperial on there, and you have your centimeters and your millimeters, that's how big a frog egg is. So you can see with your naked eye, right? You can see 1 millimeter. That's roughly about the size. Human eggs are smaller. So if somebody is undergoing in vitro fertilization, IVF, if they're undergoing those fertility treatments, that's about the size of a human egg. So now we are down to micrometers. These are micrometers. These are nanometers. So human eggs are small. Animal cells, in general, you're not going to see animal cells with the naked eye. They are, in general, too small to see with the naked eye. But here is just to give us some reference points. Here's our generic eukaryotic cell, plant and animal, and they are anywhere between 10 and 100. These are, in general, our eukaryotic cells, anywhere from 10 micrometers to 100 micrometers in size. Within the eukaryotic cell, we're going to talk about all the parts: nucleus, ER, rough ER, and smooth ER, and mitochondria. Here is that organelle, the mitochondria, and it's about 1 micrometer. Now we get down to generic size of bacteria. Bacteria's generic size, and again, they change in shape and size. They're going to have some teeny tiny ones. They're going to have some relatively big ones. But in general, bacteria are about 100 nanometers. So much, much smaller than our eukaryotic cells that were over here. So here's our prokaryote. Here's our eukaryotes. In general, prokaryotes are very small. Eukaryotes are bigger. And we saw that in that previous picture of the light microscope versus the electron microscope. And we could see the scanning electron microscope with the yellow colored eukaryotic cell and those salmonella trying to sneak in. Gives us, again, reinforcing that sense of size, that bacteria, prokaryotes, bacteria and archaea are small, and eukaryotic cells in general are larger. And size for bacteria and prokaryotes in general is smaller because they don't have all the organelles that eukaryotic cells do. We are going to talk about in the next video all the different things that eukaryotic cells. We have membrane-bound organelles like a nucleus, like mitochondria, and those things have a job. We talk about anatomy and physiology's form and function, and those structures help highly organize the cell, and those structures within the cell have designated duties, designated jobs. And prokaryotes don't have all of those highly organized designated duties and jobs inside of their cells. So they're very limited in what they can do. And so those cells are smaller. They are smaller because they cannot. If they were any bigger, they'd need to specialize into specialized organelles like eukaryotic cells have. So prokaryotes are small. They have very primitive methods of doing what they need to do, but they can absolutely do everything that they need to do. They can do all the properties of life in their little teeny tiny prokaryotic cells that are very small versus our larger eukaryotic cells that are more complicated. And we're going to go in those in the next video. So if you have questions about the first two sections, let me know. Have a great day.